From the makers of vet-recommended Greenies dental treats comes an easy way to support your dog's wellness. Greenies Smart Essentials Dry Food delivers complete balanced nutrition with science-backed recipes. Greenies supplements, developed by a team of PhD animal nutritionists, offers options to help support healthy joints, digestion, and more. Whether it's mealtime or mobility support, Greenies helps you care for your dog from the inside out. Learn more at greenies.com.
Welcome to Veterinary Vertex, the AVMA Journal's podcast, where we delve into the behind-the-scenes look with manuscript authors. I'm Editor-in-Chief Lisa Fortier, joined by the fabulous Associate Editor Sarah Wright. Today we're discussing Bumped-kinase inhibitors in horses for the prevention and treatment of EPM, which is equine protozoan myelitis, with author Isabella Rocha. Hey Isabella, thanks for being here with us today to talk about my favorite species, the horse.
Thank you for the opportunity to discuss this research.
We're so happy to have you here today. Thanks for joining us. So let's get started. So before we dive in, could you share a little bit about your background and what brought you to EPM research?
So since early when I started vet school, I became very interested in horses and neurology in general. I had an opportunity to attend one of Steve's lectures on EPM when he visited Brazil. And that's when I knew that's what I wanted to work on, being able to put together horses and neurology.
Isabella, your AJVR article discusses the potential utility of Bumped-kinase inhibitors for the prevention and treatment of EPM. What's the background on this research?
So historically, protein kinase inhibitors were considered for therapy of parasitic diseases, but the challenge of specific inhibition of parasite kinases versus the mammalian kinases had limited the practical application. So in the case of the bumped-kinase inhibitors, they were originally generated to build specificity into this drug class to compete with the ATP for the kinase active site. The bump on the BKIs precludes there to bind to almost all mammalian kinases because those have a bulky gatekeeper residue in the ATP binding pocket. So West's lab at the University of Washington were able to determine that the apicomplexan parasites have a naturally occurring small gatekeeper residue in their ATP binding site. And that is on a protein called calcium-dependent protein kinase 1 that we call CDPK1. This is a protein important for parasite motility, host cell invasion, and egress. Considering that, they embarked on a medical chemistry project to optimize BKIs for use against several apicomplexan species, including Sarcocystis neurona.
Yeah, really thorough answer. So you answered a lot of my follow-up questions, but does the biology of Sarcocystis neurona create challenges or provide opportunities for BKIs?
Yes, Sarcocystis neurona is a single-celled eukaryotic parasite. This means that it relies on many metabolic pathways shared with mammals. And that is a challenge to identify selective drug targets. Thankfully, this parasite also has many unique features that are not shared with mammals, such as what we call the apical complex, which is involved in the process of motility and invasion of the host cells, and it also has a unique organelle called the apicoplast. That is a vestigial non-photosynthetic plastid, and that's involved in fatty acid biosynthesis. So the presence of these unique structures, they're promising for a target in these parasites.
So Isabella, what are the key take-home messages you hope veterinarians will remember?
I believe this research is a great example of how foundational studies in parasite biology inform translational research. Without sequencing the Sarcocystis neurona genome and a lot of the in vitro studies, the BKIs wouldn't have advanced to pharmacokinetic studies in horses. So foundational study may take a long time to lead to the clinical applications, but it often leads to the great advancements in our field.
Isabella, are there concerns around drug resistance with BKIs like there are with other antiparasitic treatments?
So it's interesting because in the Sarcocystis neurona life cycle, horses, they are dead-end hosts in which only a sexual reproduction occurs. This means that the use of the drugs in the parasites are unlikely to lead into the resistance. That means that infection is not transmitted from one horse to another. So even if the resistance occurs at the individual horse, it's not going to become widespread. And we need to consider that the parasites, they replicate very slowly over a course of many days. So they're very careful when they are multiplying and dealing with their genome. So mutations are unlikely to occur in these parasites. So even if it were at the horse level, we think it's unlikely that resistance would occur, which is very interesting considering the biology.
Yeah, very cool. You talked about this or touched on a little bit earlier, but maybe a little bit more. Do BKI show promise against other equine pathogens or even human diseases caused by related parasites?
Yes, absolutely. So in horses, the BKIs are also effective against Babesia cavalli and Theileria equi, which are the causes of equine piroplasmosis. And this drug class is also being investigated for the treatment of toxoplasma-associated abortion in sheep, toxoplasmosis, and cryptosporidiosis in humans. So it's an example of One Health research for sure.
Yeah, I love that One Health tie-in. Thanks for providing that. And looking ahead, what are the next steps for research in this area?
So the current lead compound, BKI 1708, has a great systemic distribution in horses, but it has low CNS penetration. So it's a very promising compound for prophylaxis as it can control systemic infection, but it may not be suitable for EPM treatment at this point. So after we published this paper, we started some preliminary work with a different lead called BKI 1553, and so far we were able to show very good drug levels on CSF, which means that this compound might be more suitable for treatment than what we have studied so far. So hopefully, with more PK experiments, this other lead compound could potentially advance to a clinical trial in horses with EPM.
Very cool. And how do you envision collaborating with clinicians or other researchers to advance this One Health work?
So the main challenge with EPM clinical research is that we do not have a robust experimental infection model. That means that we need to work with naturally occurring EPM cases, and for that we need to work with the clinicians. The main challenge is that less than 1% of the horses exposed to sarcocysts develop EPM. So it takes a long time to achieve a good sample size in a clinical trial. And we also struggle with testing the efficacy of prophylactic approaches. So working with the clinicians, not only in recruiting horses for these types of study, but also brainstorming ways that we could assess how prophylactic approaches help horses. That is certainly very important.
And how might future findings translate into practical changes in day-to-day veterinary practice?
So we hope that the BKIs would eventually lead to a commercial product, which would provide a third treatment option for EPM. This would be especially important for treating horses that either do not respond well to the current antiprotozoals or animals that tend to relapse after treatment completion.
Isabella, let's talk a little bit about your journey. How did your training, all the way from interest in becoming a veterinarian and your previous work, prepare you to come up with this idea, implement the study, get across the finish line, all the way through publication and write this article?
So it's been, as I mentioned, since vet school, I became interested in neurologic diseases of horses. And it was especially for me during residency that I started treating the neurologic cases, and that became clear that compared to all other diseases that we see in horses, the neurologic diseases are the most challenging ones. And even in the case of EPM, which is a disease we know a lot about how to diagnose and treat, there is still shortcomings with that. So it was not only my interest in equine neurology, but also the day-to-day struggle as a clinician that led me to pursue training in research in this area. And it was very interesting for me as I was studying the success rate and the relapse rate for the current treatments for EPM, and they are about the same for the different drug classes that we had. So trying to develop a new product that had the potential to have a superior successful rate in horses, that was very appealing to me.
Yeah, it's surely a frustrating disease, no shortage of experiments that need to be done. And it is interesting that we can't really replicate it in a clinical model.
Yes.
Yeah, certainly hampers progress.
Yes.
Were there any mentors or colleagues that played a pivotal role in guiding your thinking or these research methods in this article?
Absolutely. So both Steve and Dan were great. So Steve's just the amazing neurologist, and he's been a great mentor on the clinical side of this research, and he's the person I look up to as a clinician. But also my PhD mentor, Dan, he's also for me the role model as a scientist. So it's very interesting because as a clinician, as we're trying to diagnose an animal, we try to gather evidence to support a diagnosis as we go through our differential diagnosis. But as a scientist, it's a little bit of the opposite as we try to prove ourselves wrong before we move forward. So we want to do a great number of experiments and test all the possibilities before we can say, yes, this is the direction we need to forward. So it's learning to think in different ways and blending being a clinician and a scientist is certainly being very interesting. And I'm grateful to my mentors for that.
Now, before we wrap up, let's have a little bit of fun. So we have 2 fun questions for you today, Isabella. First one is about communicating with animals. So if you could instantly communicate with any one species, which would you pick and what's the first thing you would ask them?
I would pick dogs, and the first thing I would ask is what do they dream about? Sometimes it just looks like they're having fun, and I wonder if they're just reliving their day or prepping for the next day, and but also knowing what their nightmares look like, what they have to worry about so much that they have nightmares.
I like the dreaming one. I'm not so sure I want to know about the nightmares. Maybe I petted them too fast, came at them too fast, didn't let them out enough. On on the human side, if you can instantly master any skill related to patient care behavior, what would you choose?
Certainly, the spinal taps. As Steve likes to say, whoever says they never struggled with a spinal tap haven't done enough spinal taps. Every once in a while, we get a horse that is a little bit crooked in their back, or we're just not having a good day. So I like to think that spinal taps, they're half technique and half luck. You never know.
That's awesome. True.
That's a very good answer. Well, Isabella, thank you so much for joining us. We appreciate you being with us today and for sharing your work too with AJVR.
Thank you very much for the opportunity to share this research. It's very exciting to me, and I'm grateful for being able to talk more about TPM, which I'm fascinated about.
And for our listeners and viewers, you can read Isabella's article on AJVR. I'm Sarah Wright here with Lisa Fortier. Be sure to tune in next week for another episode of Veterinary Vertex. And don't forget to leave us a rating and review on Apple Podcasts or wherever you listen.